Excellent. So yeah, my name is Aniruddha Agnihotri. I'm originally from India. You guys can call me Annie. I started my PhD here in Perth at Murdoch University in Dr. Vezu's lab in 2017. I'm almost about to complete my PhD now. I'm waiting for my thesis to be corrected. So today's topic of my presentation is unfolding the behind refolding. Although the title looks a bit confusing and fancy, but the original motive behind the presentation is I work on odorant binding proteins, which are basically chemosensory proteins. So if we try to see at a glance the insect chemosensory system, there are different components involved into it. Uh, some of them are membrane bound proteins like gustatory receptor, olfactory receptor, and some of them are soluble proteins, which basically all of them work together to generate the chemosensory signals. My today's presentation is focused on odor and binding proteins. So basically, the question is, what does odor and binding proteins do? Odor and binding proteins are present in different chemosensory organs of the insects. Right now, let's consider an antenna. An antenna has different types of sensillas on top of them. These sensillas are nothing but projections, and these projections has neurons within them. These sensory neurons are, of course, surrounded by sensory lymph, which is hydrophilic media against it around it. So these proteins are present in this sensory lymph. What is the use of these proteins? Basically, the volatile ligands that is present outside in the environment, this protein fish those volatile ligands and transport it to the membrane-bound receptor, which is odorant receptor, onto the neuron, and that's how these volatile ligands go and bind to the receptor, and then the downstream signaling process starts. So these are transport proteins. If we Why is it necessary to study the refolding of odor-binding protein? Now, before we go that, just briefly talking about refolding. Refolding is one of the way to produce in recombinant proteins, especially if you try to express them in a bacterial host like E. coli. Either your protein might be expressed like a soluble form, or it might get expressed like in an insoluble form, which is usually called as inclusion bodies. And these inclusion bodies are they do not have the structure, so you need to refold them in vitro after removing them from the bacterial cell. If we try to have a look from for last 10 years, the amount of research that has happened on this protein, it looks like more than 50% of the research, and the data is taken from PubMed and Google Scholar, is on the recombinant expression and characterization of these proteins. So that's why when I came here and I started studying this protein, I came across this huge set of data which only talks about recombinant expression. But unfortunately, very few articles really go into that detail and try to study how the recombinant expression is happening. If they're doing a refolding, are they doing in a correct way or not? So before going towards the actual experimental data, uh, the just briefly telling you about the host system and the model system that was chosen here. So the odorant binding proteins that we were studying was of Helicore parmigera. It's a lepidopteran pest, highly polyphagous, and there are more than 50 different OBPs that are already been identified and studied till now. So this data was previously done in my lab, and we found out, in my lab it was found out like around 46 odorant binding proteins, and they found out different expression of them in either in tissue or the life stages form. And out of this, we decided to go ahead with OBP2, OBP5, and GOBP2. We initially decided to go ahead with 6 OBPs, but then here I'm just showing you the data of 3 OBPs. And these OBPs, if you see about OBP2, it is specifically expressed in male darsus and female darsus, whereas OBP5 is also expressed in the same chemosensory organ. Whereas GOBP2 is mostly present in antenna. So we decided to focus on these 3 odorant binding proteins. So the strategy was here to express them in an E. coli host using a bacterial vector, which is PET30A vector here. The proteins were expressed along with a fusion partner, which is a 6-histidine tag. The motive of having a tag to to the protein was to basically use it like a tag to fish out and do a purification using affinity chromatography. So the host cell that was chosen was E. coli BL21D3 competent cells. Of course, when we express this protein, they all went into inclusion bodies. I have a result for it ahead. But before that, I will talk about when we express this protein and when we got the inclusion bodies in our hand, which were the different refolding methods that I actually chose here to study. So if you see, there are 5 different refolding methods that I'm showing you here, out of which the last one, which is ACR, which it was developed by me in the lab, which is refolding by acidification, whereas all these first 4 methods are already well-established methods that are commonly used by different protein biochemists to refold different proteins in vitro. The reason why we decided to do a comparative analysis with these 4 different methods was these are the most popular methods also used for OBP refolding, and all the literature that I got, they have been used a lot. And we thought, okay, it's very important to compare and to study which of these methods are really good and what are the challenges associated with each method. So to just talk about comparative refolding, when we expressed our protein, this gel shows us that it was mostly expressed in insoluble form. So these are only insoluble fractions that was loaded onto the gel, and most of the expression was in insoluble form. Initially, when I started doing the refolding with all these 5 methods, I realized that uh, the recovery of the protein after refolding. So when you refold your protein, the immediate recovery is in the form of soluble form. If your protein is not soluble, it is precipitated out, it is a bad sign. It means that the protein was not folded correctly. So you can see here that the best method that was seen here was mostly my method, which is ACR, redox method, and the normal dilution method, NDR. Whereas the refolding by dialysis and refolding by on column was not that good for all 3 OBPs. So we decided to only focus on these 3 methods here. After the refolding, we decided to analyze the refolded protein using a reverse phase HPLC chromatography. Basically, it was to see whether what is the heterogeneity of the protein mixture there. When I say heterogeneity, it means that when you refold your protein, if your protein is not precipitated out and if it is soluble, it doesn't really mean that it is all good. What it means that it might be either in a soluble aggregated form, or it might be in a monomer good shape, which is closest to the native shape. So we are of course interested in a soluble monomer form. So in this reverse phase data, you can clearly see that these big ugly peaks, plateau peaks, are representing the aggregated form of this protein, which is a soluble aggregate. So you cannot see it because it's not precipitated, but still it's in a bad shape. Whereas this sharp peak only represents a monomer form. So what it tells that if you just blindly refold a protein, you cannot really trust on it unless and until you do a systematic analysis of it. And this sort of analysis is unfortunately absent in most of the published data about odorant binding proteins. Same goes with OBP5 as well and GOBP2 as well. Uh, uh, I did the analysis not only after refolding, but I also did it after post-purification, which means that I used an affinity chromatography technique to purify this protein after refolding, because I told you earlier that it has a his tag attached to it. So using that his tag, I purified it and also checked it after the purification, what is the state of it. So later, to further characterize this protein and to see if so based on reverse phase analysis, it was understood that my method, which is the acidification refolding method, and the redox method are the best method, which even passed through the reverse phase milestone, I would say. And now we have to see whether out of these 2 methods, what is the difference between the refolded protein using these 2 methods. To check that, this protein has a very peculiar structural characteristic, which is 6 cysteine residues in their sequences. Those are like signature motifs for them, and they form a very specific disulfide linkages, which basically stabilizes the entire structure of the protein. So I decided to do a disulfide mapping experiment. So what you do is you cleave a protein using a protease into different fragments, and you try to see whether the disulfide linkages that are formed are whether they are formed or not, first of all, and if they are formed, are they in a correct order. Like for example, if first cysteine has to be paired with C3. Is it really pairing with C3, or is it pairing with C2 or C4 or C5? So basically, that's what a disulfide mapping really means to the. So when we check the data, it can for OBP2, you can see that this data is basically, this is how do you do it? You cleave the protein using a protease, and then you put it onto a MALDI, which is an MS based method. And on MALDI, you try to see the intact mass of each fragments, and those who are the fragments which are connected by disulfide linkages, they will have a combined mass, whereas if they are not connected by disulfide linkages, they will show an independent peak. So that's what is seen here. So this is the typical pairing of the disulfide linkages here. C1 pairs with C3, C4 pairs with C6, and C2 pairs with C5, which is all present in my in a OBP refolded by acidification method. Whereas you can clearly see that in case of redox method, there are signs of free cysteines, which means that the disulfides are not formed there, which is C5 and C2 and C4. Those were not clicked with each other, which means that in this method, although it was monomer and it was all looking good even on HPLC data, but when we go to at a structural level, it looks like the disulfides are not formed. So which means that this is more unstable, and this is prone to misfold and basically aggregate, whereas this has actually gone to closest to the native structure, and this is a good structure that we can attain after refolding. Similar data was also obtained for OBP5 and GOBP2. Due to time constraints, I'm not going into details, but basically for all three proteins, it can be seen that disulfide pairing through disulfide pairing, we understood that acidification is the best method even at disulfide linkages formation. Just quickly talking about after how did I the major challenge in most of the recombinant expression is that you have to choose multiple chromatography methods to purify it. Just one purification round is not enough, so you have to do. And one of the most commonly used method is size exclusion FPLC, which is basically purifying your protein based on sizes. The disadvantage of size exclusion FPLC is the instrument is expensive, the columns are very expensive, and it's a very time consuming method. It takes almost a day to prepare your buffers and do and especially if you are doing at a big lot. So there is another method in literature which is very well used, which is not used for purification, but it is used to either precipitate a protein and to concentrate it, or to buffer exchange it, which is ammonium sulfate based precipitation. It's a very common method to precipitate protein. But I realized a fact here that if you precipitate a protein using different concentrations of ammonium sulfate, which is like lower percentage and higher percentage, lower percentage usually precipitates only the misfolded or aggregated proteins, whereas higher percentage, or the percentage that is actually suitable for the good soluble monomer protein, will remain soluble and will remain in a solution even at lower percentage. So it turned out to be like a purification method. So what I did is that the OBPs that were refolded by my method was precipitated first at lower percentage of ammonium sulfate, then it only resulted into precipitation of aggregates and misfolded proteins, and then the soluble monomer or the good protein was then precipitated again with higher percentage of ammonium sulfate, which resulted into concentrations of this protein. And this all was tracked on HPLC, and it can see that as you go ahead, your digestion means that you remove the his tag, the fusion partner, which has to be removed when the purification is done. So this is the refolding stage. Then this is the first purification and digestion, that is removing of the his tag, and this is the second purification, which is basically the ammonium sulfate purification. And that you can see that the protein is very clean without using a second chromatography method, and which is, I think, a big advantage here. Just quickly talking about the last part of my data. So basically, this was all about refolding, but the main motive of doing a refolding study was to check whether if this change in structure, or if we ignore the refolding success, will it also impact the binding efficiency or the functional characterization of this protein. So to do that, I developed a new method based on reverse chemical ecology. So this is a setup that was developed, and you can see that these proteins fish out the volatile ligands released by the plants or the environment, and they they bind these ligands and transport it to the receptor. So what I did is I used a buffer PBS in which the protein was kept, and a plant was kept in an airtight chamber, and a compressed industrial grade air was passed through the chamber, let it mix with the plant volatiles, and the volatiles were then transported to the air and mixed with the odor and binding protein solution here constantly. And then hoping that the native ligands or the ligands that can bind to this protein will bind and the remaining would not bind. So later on, this bound and unbound ligands were separated out using solvents such as ethanol or diethyl ether or other organic methanol or these organic solvents. And basically they were depending upon the whether you want to choose a polar solvent or a non-polar solvent. And then these proteins or these bound and unbound ligands were characterized using a GCMS data. And on GCMS data, you can see that there are different ligands that was found to be binding to the protein here. So it's not just one ligand, and this has already been known into literature that one OBP does not necessarily binds to one ligand. It can bind to numerous ligands or to numerous compounds. And similar scene was also seen here, whereas in the control, which is like without a protein and just buffer, and the second was intentionally chosen a different protein just to see if that makes a difference, and that was lysozyme, and it was clearly not binding to anything. So only odor and binding protein was binding here. Then these ligands based on the heat map that we generated up onto the based on the peak area data, we found out that there are top 4 ligands that are showing highest binding with our protein, and these 4 ligands were then used to do a computational study of binding with the protein. So for that we modeled our protein, and then modeling where we found the core of the protein or the binding site of the protein, and we found out, okay, these ligands have a good binding affinity with the protein. But out of that, we just ranked it based on the binding affinity, and it found out that each protein shows some preferences with the ligand. Similar data was also obtained for OBP5 and GOBP2, and for each protein we identified some top 4 ligands. Out of the top 4, we ranked them based on the binding affinity, and we found out that each protein has one ligand that has highest affinity to it. For example, OBP2 has 2,3,8-trimethyldecaine, OBP5 has D-limonene, and GOBP2 has 2-cyclohexyl-undecane. And for each of these ligands, there was some literature support available. For example, especially OBP5 was really surprising. There was a recent paper published early this year, which talks about olfactory exposure of Helicoverpa armigera. Just a second. Olfactory exposure of Helicoverpa armigera larvae to limonene dramatically increases larval tolerance to carbamate insecticide. So all these ligands were also supported by the literature or the previously done research. We aim to do some verification study based on using other different binding assays, for example fluorescent based binding assays in future, which is still going on in our lab. So in conclusion, we just this whole was to prove that you cannot ignore the analysis of refolding, protein refolding, if you are using that as a recombinant expression method here. And in most of the literature, that's what is done. So it was shocking to us, and we thought, okay, we should address that point, and that's what we did it. So thank you. This is my group, and if anyone has any questions, I'm happy to answer it.
All right, thank you, Aniruddha. I saw we had some extra time to go over, so I didn't stop you, but we still have 5 minutes until closing remarks, and if there are any questions, we do have time.
Oh, I was too fast, I think. Huh?
Sorry, that was some of the most information per minute I've ever seen in a talk, so I kind of forgive you. But yeah, any other questions before we go to the concluding remarks?
Is it on chat? Oh, no.
All right. Well, I'll give everyone a little bit of time to take a quick break and get to that first Zoom link, and Aniruddha as well. But people can feel free to email you, send you any questions that they have, right?
Sure, of course. Yeah, definitely.
Yeah.
Thank you.